All right, welcome. Welcome to the FARA Flash Talks. I am Liz Sorani, Director of Research at FARA. And to celebrate FA Awareness Month, which is the month of May, we want to bring you some exciting research in FA during this month. Every Thursday at noon Eastern Time, you can tune in to hear from several junior investigators from all over the world who will share with us the important findings of their research in FA. We really hope that this is an opportunity to learn more about what type of research FARA funds. As a lot of what you'll be hearing, most of it is funded by FARA. And also a great opportunity to meet these young investigators and to encourage them to continue to work on FA during their careers and continue to make a great contribution to the FA research. So the presenters only have 5 minutes to share their research, and this is a challenge for them to summarize their work of months and sometimes years in such little time. And it's also a challenge for us who are attending, listening in, to appreciate their take-home messages and the relevance of their research. So really, let's take this opportunity to ask questions and clarifications. We have 5 minutes after each talk for questions and to discuss their work with them. You can type your questions in the Q&A box at the bottom of your screen. Like last year, we're also asking you to rate the presentations with a very short survey at the end of the webinar. So keep your browser open at the end and you'll be prompted to fill out the survey. And we will recognize the most voted presentations with a little award. Don't worry if you cannot attend all webinars. We hope you will, but if you can't, we plan to post all sessions on YouTube. There it is. Of course, COVID had to have its say on our plans, and one of our presenters today, unfortunately, Zakaline Suleiman from Brunel, is sick, and we wish him a speedy recovery, but he plans to present at a later date. So we only have 4 speakers today. Now we can get started, and I'm happy to leave you in the capable hands of our moderator today, Jessica Hawley. We'll make sure that everybody sticks to their time and who will take questions at the end. I will now let Jessica introduce herself first. Thank you, Jessica.
Hi, everyone. As she said, my name is Jessica. I was diagnosed with FA at 23, so about 6 years ago now. So all this is very exciting for me. Kind of jumped in headfirst this last year and a half in becoming an ambassador with FARA, working with the Every Life Foundation, and really anything that I can do to kind of help progress the research that we need here in FARA without having a really big scientific background. So we're gonna go ahead and get started. Up first, we have Hughster Almost from the University of Buenos Aires, and he's gonna speak to us about the learning about function of Friedreich's ataxia from the amoeba.
So, does it look good? Everyone can hear me?
Yeah.
Okay. So, well, my name is Hughster Almost. I'm a biology student here at the University of Buenos Aires in Argentina, and I'm currently working at the IB3 Institute together with Javier Santos' team. Today I will be presenting my work titled Learning about the Function of Frataxin from Amoebas. Well, our team has been recently looking into proposing Dictyostelium discoideum, which is this little amoeba here, as a model organism for the study of Friedreich's ataxia. Model organisms are organisms which have been thoroughly described in their biology, and for which experiments and methods are usually very well optimized. And the usefulness of such organisms lies in the fact that when they are correctly chosen for a given biological phenomenon, any discoveries made on these organisms can provide useful insight into other species like ourselves. So in order to validate Dictyostelium discoideum, which we like to call Dicty, as a model organism for FA, we would have to prove that the molecular mechanisms which underlie this disease are also present in this amoeba. In other words, it would mean to show that Dicty has a mitochondrial supercomplex, which is similar to the one related to FA in humans. And in order to do so, the first thing we did was to evaluate if Dicty has equivalent proteins for all of the human-related FA proteins. Here I am showing, focusing on these 5. And we found out that Dicty has genes which are very similar to all of these 5 proteins and many others, which suggest that Dicty does have a mitochondrial supercomplex and that it is made up of the same proteins as it is in humans. Then we purified Dicty's frataxin, which is here represented as DD-frataxin, which is a very important protein in this disease. We purified it in our lab and wanted to see if this frataxin does work similarly to how it works, to how human frataxin works. On one hand, we tested if DD-frataxin is able to bind to the human supercomplex. If this was the case, it would mean that the physical interactions that allow this complementation are somewhat preserved between species. And we found out that Dicty's frataxin does in fact bind to the human supercomplex. It does not bind as strongly as human frataxin, which of course was to be expected since they are different species, but it binds very strongly, especially considering how different humans and amoebas are, of course. On the other hand, we wanted to see if Dicty's frataxin, through this interaction, was able to activate the human supercomplex. This activity here is represented as transformation from A to B, B being the product of this activity. We tested this and found out that Dicty's frataxin is also able to activate the human supercomplex. Here, this activity is represented as 100% for complementation by human frataxin. 23% would be the activity without any frataxin, and 63% is the activity we measured by complementation with Dicty's frataxin. Well, these results would show that Dicty's frataxin is not only able to interact with its respective supercomplex in a similar way it does in humans, but that this interaction allows for this activation also in a similar way. So binding and activation will be preserved between species. So these results, together with many other experiments my colleagues are working on, are helping us build Dicty as a future tool for the study of Friedreich's ataxia, and will hopefully one day become a useful model organisms in which to test new treatments or scan for drugs and a lot of other things. So that will be all. I hope I made myself clear, and I hope you enjoyed it.
We're really on time there. That was officially 5 minutes, so good job.
I'm happy to hear
If
it.
we have any questions, go ahead and put them in the Q&A. I'll give you guys a few minutes to do so.
Yes, I will stop sharing now to see these questions. So
the question that has come in is that can you reduce for FXN with DT?
well, I'm not sure what do you mean by reduce product or produce will be reduced like in a chemical way.
Yeah, maybe you can, can you like use something like siRNA, or can you knock out frataxin? I think there's
Yes.
another question, you see the effect of knockout or knockdown of frataxin. Is it
Well,
essential like it is in human or in mammalian cells?
well,
She
those are
did
great questions.
clarify. Oh, sorry. She
Yes,
did
yes.
clarify asking genetically or others.
Yes, yes, yes. That's a great question we have made ourselves also. Well, even though I'm particularly focusing on the more biophysical properties of these proteins, a lot of other presenters of our team, which we'll be presenting later in other days, are proving knocking down frataxin in DICT and knocking out. It is possible, and it is also quite easier than it is in human cells, because DICT is does only have one copy of each gene, not unlike us, which have two. So it is quite easy to try to knock out or knock down these genes, and it is possible.
The next question we have is, what effects are seen with KO or KD of DT for taxin?
Well, some of the results we have is that well, DICT has a very complex life cycle with a lot of phases, and knocking down or knocking out these frataxins does have proved to reduce its capacity to enter in new phases of this life cycle. It also has a very slow growth rate, and many other, well, phenotypical changes we are trying to measure.
We also have a question of what are the shortfalls of using DICT to model FA?
What are the, sorry?
The shortfalls.
Oh, so yes. Well, it would mean that I will say it is obviously a very different organism. Amoebas are very far evolutionary speaking from us. However, DICT does have a lot of background as being used as a model organism for the study of many mitochondria-related diseases in humans. So it has proven itself to be a useful model in the past.
So we only have time for one more question. There are a couple others, but we'll have those answered in the chat. So the last question we're going to ask live is, does the amoeba growth in normal O2 or hypoxia?
Well, this amoeba usually grows in normal O2. It's found all around the world in different forests and rainforests. And we haven't tried to growing it in hypoxia. I don't believe it can, but you will be listening more about it in later presentations. And many of my colleagues have a lot more expertise in this field, so they will answer these questions more precisely.
Thank you so much for your presentation. We do have a couple more questions in the chat if you couldn't answer. And then if any other come in for you, I'll go ahead and let you know. I
will be answering them. Thank you.
Next up, we're going to be talking to Deepika Mokakemi Philippandi. I'm really sorry if I mispronounced that. She's from the Institute of Myogene, and she's going to be speaking to discovering new disease pathways in FA brain cells.
Thank you, Jessica. Hi, I'm going to first share my screen. Just give me a minute. Okay, you all see my presentation?
I don't see it.
You don't see them?
I don't know if it's a problem on my end.
No, I think.
No, it says that you're sharing your screen, but your slide wasn't showing up.
Okay, I will see. Okay. I think you can. No?
There it is. Yeah.
Oh, perfect. Okay, that's good. Okay. Hi, everyone. I'm Deepika Mokchami Chalapandi. I'm a second-year PhD student working in Helen Puccios laboratory in Lyon, France. And today I'm going to present to you the discovering new disease pathways in FA sensory cells. In FA, one of the primary affected tissues are the sensory neurons, and these sensory neurons are found in a tissue called dorsal root ganglia, which we will refer to as DRG from now on. So DRG is made up of several types of cells. There are a lot of neuronal cells and non-neuronal cells, and for the relevance of the study, I'm going to focus on the three major neuronal types found in DRG. Those are nociceptors, mechanoreceptors, and proprioceptors. In FA, we know that proprioceptors are one of the early and severely affected sensory neurons, and the sensory, the axonal degeneration and the neuronal degeneration of proprioceptive neurons is severe compared to other sensory neurons. So the function of these proprioceptive neurons is called proprioception. Proprioception is the ability of the body to sense its movements and location in space. So in FA, as it's been already reported that there is an early degeneration compared to other neurons. There is no existing elaborate knowledge about this phenotype. This leads to my research question that what makes these proprioceptive neurons more susceptible to loss of frataxin? What makes them sensitive? To answer this question, as a first step, I decided to study the genetic signature of the proprioceptive neurons. Genetic signature of a cell is what determines the cell's function and specification in a tissue. Now you can imagine different types of cells as different types of fruits, for example. They have different colors, different flavors that has been determined by their own genetic signature. So to understand the genetic background of the cell, in my case, the proprioceptive neurons, I will use the techniques called transcriptomic approach. So now you can imagine this plate of fruits as DRG with different types of cells. And there are two transcriptomic approaches that can be done. One is called the bulk RNA sequencing. So bulk RNA sequencing is the part where you put everything together and then you extract the genetic background of the different types of cells. The downside of this technique is that you get all the genetic background, but you will not be able to specify the genetic background of different types of cells individually. They will lose their individuality even though you have the genetic background. So this is a very effective technique, but this has a downstage. So to overcome this problem, we use another technique called single cell RNA sequencing. So in the single cell RNA sequencing, you'll be able to see the genetic background or genetic makeup of each and every cell, which is more useful in terms of understanding the genetic background of a particular cell type, in our case, the proprioceptive neurons. So in the graph where you see different clouds of little colored dots is basically different types of cells that are being isolated based on their genetic background. And this is a very effective technique that will help us to study the proprioceptive neurons and understand what is happening in a molecular level in this neurons when there is a loss of frataxin. So now that I have a technique to study my cells, I need a mouse model that actually that replicates FA like phenotypes. To do so, we use a neuronal mouse model in which the frataxin is deleted, particularly in proprioceptive neurons. This conditional knockout mice is a well-characterized neuronal model that can help us study the molecular background of proprioceptive neurons. And once you have the mouse model, all you do is you extract this tissue called DRG from the mouse models, you process them through single-cell RNA sequencing. And once you have the data from this experiment, what we do here is that we collect the list of genes that are expressed in proprioceptive neurons. So now that I have a list of genes, to actually know which genes are changed or which genes are highly dysregulated, we compare them with the control DRG genes, gene expression levels. So comparing the control genes and the control mouse model and the disease mouse model gives us the levels of dysregulations of genes. And based on the level of dysregulation, the highly dysregulated genes are selected and they are processed in a specific gene ontology method that will tell us the specified pathways of these genes where they are involved. And for identifying these pathways.
I'm so sorry, Deepika, I've got to cut you off. We are at 5 minutes.
Okay, I'm just gonna finish it off with the perspectives or no?
No, you got to stick to your 5 minutes. I'm so sorry.
Okay, thank you.
And then we will go ahead and take some questions.
Yeah.
The first question is, did you see any transgressional stress response get turned on in the SC sequence? For example, NRF2 signaling or the ingraded stress response, or any stress response not linked to the FXN dysregulation?
So far we don't have a lot of details on the data because the data analysis is an ongoing work right now. So we have optimized the protocol to do the single cell RNA sequencing, but the data analysis is still on process. We don't have the information on dysregulated pathways, but we can expect them dysregulated.
Our second question is, would you expect the clustering of proprioceptive neurons to change when compared at pre and post developmental stages? So the clustering of proprioceptive neurons are based on their genetic makeup. It differs in embryonic stage compared to the adult mice. So I'm currently working with adult mice and I have not handled the embryonic data, but there are different genes, biomarkers expressed in different stages. So it makes the clustering different. Okay. Will the SC sequence data identify the cell tag clearly?
So far, yes, you can actually you can select the biomarkers that are specific to your cell types that will make more clusters. So it's a question of identifying major types of neurons to their subtypes. For major types of neuron clustering differentiation, they can identify it. But for the subpopulation, there might be struggles or struggles on clustering them. Might be, I don't know. But so far, clustering has been good and we were able to identify all the different types of cells.
Okay, it looks like we do have one more question. In the mouse model, we'll have deficiency of proprioceptive neurons. So most of the rcRNA sequence data will likely be from another DRD neuron.
Yes, it's so I don't know if I got the question right.
So it's asking if, because in FA you'll have low frataxin. With low frataxin, you have deficiency of proprioceptive neurons. If in the mouse, you have mostly other neurons when you extract your RNA from the DRGs. Because the proprioceptive neurons will be at that point, will be either dead or, defective.
Yeah, so that's a very good question, because we use two different time points to determine the genetic background of the proprioceptive neurons. One is the early stage of the initiation of the disease, and the other stage is when the axons are completely degenerated. So in both the cases, we were able to retrieve the cell body, and we were able to retrieve proprioceptive neurons. So in mouse model, even though this is not even a low level of frataxin, it's a complete deletion, it's a severe model, we were able to have two stages in which we were able to retrieve proprioceptive neurons.
Wonderful, thank you. There is one more question in the Q&A. If you could go ahead and answer that on chat. Sure. We're gonna go ahead and move on to our next presenter. So Nicole Sales from Weill Cornell Medicine will now give us some insight into investigating heart cell metabolic changes to understand FA and cardiomyopathy.
Hello, one moment. Can everyone see my slide okay?
Yeah.
Hello, everyone. Thank you, Jessica. My name is Nicole, and I am a fifth-year PhD student in Dr. Giovanni Manfredi's lab at Weill Cornell Medicine. And today I will be presenting my research, which focuses on the role of mitochondrial dysfunction in cardiomyopathy of FA mouse models. As we know, cardiomyopathy is one of the most prominent pathological features of FA, and heart failure from cardiomyopathy is often the cause of death, seen in 60% of patients with FA. And this usually occurs in the third or fourth decade of life, but surprisingly, the FA heart presents and maintains adequate function until shortly before death. So it appears that the FA heart can adapt for at least initially due to the loss of frataxin. However, eventually the heart does succumb to this frataxin loss. So there is recent evidence of stress responses in the heart in a mouse model with complete loss of frataxin levels, the frataxin knockout mouse model. And we wanted to explore the effects of reduced frataxin levels in heart in a mouse model. So we chose these three mouse models, the Kiko 700 and YG800, both have expansions, one of a human transgene and one of a single mouse allele. And then this third mouse model of the G127V, which has both frataxin genes mutated to this G127V. And interestingly, the frataxin G127V model mouse heart displayed both markers of cardiomyopathy as well as some stress responses. So we have chosen this mouse model to explore some mitochondrial dysfunction related to reduced frataxin levels, not complete loss of frataxin levels. And we believe that this mouse model presents with cardiomyopathy likely due to a very severe loss of frataxin seen by both Western blot and ELISA from this mouse model. So with this mouse model, we wanted to study the involvement of the integrated stress response, which responds to mitochondrial dysfunction. And I wanted to briefly go over the involvement of the integrated stress response in the heart. And what we've seen is it really focuses on this protein called ATF5, which has a mitochondrial targeting sequence that targets this protein to mitochondria. And when they're healthy, this protein is quickly imported and degraded. And if mitochondria are stressed, potentially due to very low levels of frataxin, then the ATF5 protein is not imported into mitochondria and is rather located to the nucleus from its nuclear localization signal. And within the nucleus, it acts as a transcription factor to alter the expression of many genes in a plethora of pathways. And these pathways together try to promote the regeneration of healthy mitochondria and heart tissue survival. And some of these pathways include mitochondrial proteostasis, which is the involvement of proteins and chaperones to help fix both proteins and structure of mitochondria. It also limits the biogenesis of mitochondria or generating new mitochondria that may also be damaged. It activates antioxidant responses to limit the damaging production of reactive oxygen species. In the heart, it has been shown that it undergoes a glycolytic shift, which shifts its substrates used for energy production from lipids to glucose. And it also has what we call like a life raft or a metabolic rewiring that again is just promoting heart survival. And in short term, these pathways are presumed to be extremely beneficial to heart function and mitochondrial function. However, the prolonged activation of these pathways may become detrimental and may influence the adaptation and then eventually demise of heart under conditions of stress. So in our model, the G127V model with severely reduced frataxin levels, we would like to analyze the integrated stress response in these hearts over time by both transcriptomics, so looking at the involvement of many of these genes in these pathways. We would also like to analyze imbalances in metabolites by metabolomics and also compare both of these big data sets to a pathological evaluation of these hearts, so correlating some of the molecular changes to the overall structural and functional changes of the heart. And together, we believe this can advance our understanding of cardiomyopathy related to frataxin level reduction. And that is my talk. Thank you.
Okay, perfect. Thank you so much, Nicole. We'll go ahead and take any questions. Okay, our first question is, what is the reason for the disparity levels of frataxin seen in the G127V mouse versus or via Western blot, almost none versus ELISA 30%?
Yes, this is a very interesting question that we now see. One possible answer is antibody detection, what the ELISA uses versus what an antibody used in Western blot what epitope it recognizes. So we are going to definitely analyze this further with the use of different antibodies and doing both Western blot and ELISA to see if maybe some epitopes due to the mutation are now not being recognized by Western blot. But this is a question we are going to address with further antibody use.
Our next question is, does ATF5 activate or influence NRF2?
Yes, so in other mouse models of cardiomyopathy, these two pathways, so NRF2 regulating antioxidant responses, have been correlated. And it is an interesting, in some mouse models, it's been interesting to observe the effect of NRF2 on heme degradation and seeing how ATF5 regulates through NRF2 heme degradation and how that might be at play in this model where you also have frataxin loss and presumably iron sulfur cluster loss as well. So yes.
Our next question, what kind of experiments do you plan to carry out concerning metabolics?
So for metabolomics, we plan on doing an untargeted mass spec of whole heart tissue from these hearts. I'm not sure if that's the question you're asking, and using, so from having, we're not going to do isotope labeling, but just steady state levels of metabolites. And through that, we will correlate with some of the pathways that regulate these metabolites from the RNA-seq.
Do you plan to use your techniques to detect when the damage is done in the mouse model's development?
So yes, right now we have two, for the G127V mouse, we have two time points that we're using. We're not looking at development right now, so early heart development. but we're looking postnatal, and we do have 2 time points, and to see, yes, when stress is activated versus when heart pathology is affected, and we would like to compare those two.
Okay, our next question is the TAS transcription a factor in the ATF5 pathway?
I'm sorry, I do not know the the answer to this question right now, but I can look into that and get back to this answer, this question.
I'm gonna go ahead and leave that one in the Q&A box so you can just get to it when you have an answer.
Thank you.
And then we do have time for one more. That is, does the YG8 mouse have a different phenotype from the Kiko? Looks like they have a similar number of GAA repeats.
In terms of cardiomyopathy and some heart pathology, we do not see differences with these 2 mouse models, even though they have similar repeats. And I think this could be an effect, how many repeats and what I think the levels of frataxin, the reduction in the levels of frataxin, and how they affect the heart are quite similar. And we just don't see a very large effect of these mouse models on heart pathology.
Okay, thank you, Nicole. I did leave that one in the Q&A for you, so you can just get to that when you have time. And then our final presentation is going to be from Anusha Siva Kumar on gene editing and stem cells, turning futuristic concepts into realistic therapies for FA.
Good morning, everyone. I hope you can see my slide. I'm Anusha, and I work at UC San Diego in Dr. Stephanie Chucky's lab. And our lab focuses on utilizing hematopoietic stem cell transplantation for therapeutic interventions with genetic disorders. And hematopoietic stem cells are cells that reside in the bone marrow, and they make the population of blood cells that circulate in the body. So we wanted to understand if we could use these hematopoietic stem cells to treat Friedreich's ataxia. And on the left, you see two studies that we did before, and on the right, you have the studies that we're doing right now. So initially, we used these hematopoietic stem cells that we collected from green fluorescence emitting mice and transplanted it into Friedreich's ataxia mice. And we were able to see that these cells lodged into the organs that are primarily affected with Friedreich's ataxia, like the spinal cord, muscle, and heart. And when these cells were there, they were able to deliver healthy frataxin. And this was also able to rescue some of the phenotype or the behavioral defects that these mice were showing. So subsequent to this, we did a second study where we wanted to see if we could do similar things with hematopoietic stem cells from Friedreich's ataxia patients. So we had healthy and patient volunteers who gave us the hematopoietic stem cells that we collected from their blood samples. And we did a experiment called CRISPR-Cas9 gene editing, where the GAA repeats that's in the frataxin gene were cut off from the gene using the CRISPR-Cas9 technology. And this restored the levels of frataxin in the CD34 samples. So now that we have two different methods where one can deliver the frataxin to the deceased organs and another one that can help the cells produce good levels of frataxin, we now transition back into my study where we do the same thing now, but now combining both of these methods. So we collect the hematopoietic stem cells from Friedreich's ataxia mice, do the CRISPR-Cas9 gene editing in the lab where we make these hematopoietic stem cells produce good amounts of frataxin and then transplant them back into another Friedreich's ataxia mice. And we look at the expression of frataxin across all of the tissues that are of primary interest with the disease. So when we did this transplant and studied the mice after 6 months of allowing them cells to move around in the body through the blood, we were able to detect these gene-edited hematopoietic stem cells in several of the tissues, and we had good levels of editing in spinal cord, which is one of the main site of the disease progression. And we were also able to see editing in the heart, brain, and also the skeletal muscle. So which is the graphs that you see on the left side. And we also were able to detect good levels of frataxin protein in all of these tissues. So now that we know we are able to deliver frataxin, edit the cells and deliver frataxin to these tissues, does this help in improving the behavior defects that we see in these mice? So we do an experiment called catwalk, where the mice walks from point A to point B on a platform, and this platform is connected to a camera. So it's going to detect how much is a difference in the way it walks, the pressure that it exerts on the surface to move from one point to another point. And it gives us a lot of parameters that we can analyze to see if there's any defect and if the edited cells that we gave them were able to restore some of these factors. So if you look at the graph on the right, you see that there's one that says number of steps, which counts the number of steps that the mice takes from moving from point A to point B. And with the mice that were progressing with the Friedreich's ataxia, they took a lot longer and more number of steps to cross the platform, whereas the edited cells had nearly equivalent to that of the normal mice. And the second graph is called as a regularity index, which tells us if there's any irregular movement in the way that the mice walks. And with the unedited mice that had the Friedreich's ataxia, there's a little less in the way that how it works, and this was almost equivalent to the healthy mice that we saw with the way that they cross the platform. And the third one is a paw print area, which tells us if the mice are relying heavily on the hind legs, or if they rely on the front legs, or if it's going to be tilted towards the left, or is it going to be tilted towards the right. And we see some differences in the way that the mice prefers to walk when they have the disease that has progressed for quite some time versus the ones that receive the edited cells, and they were almost again equivalent to the healthy wild type mice. So with all of these data, we are at the preliminary stage, and we are conducting several more studies to understand if there's any behavioral changes. But once we conclude this, what we hope to achieve from this kind of study is to collect blood samples from Friedreich's ataxia patients, do CRISPR-Cas9 editing in the lab that's going to make these cells produce frataxin and transplant them back into the same patient, and that's going to lodge into the organs, all of the organs that gets affected, and keep delivering frataxin protein throughout the lifestyle. So this is the hope of the entire project that we have been doing through the series of experiments. So that concludes my talk, and I'm happy to take questions.
Yeah, we'll go ahead and take questions now. I'll let you know as they are coming in. Okay, our first question is there a way to target stem cells so that they are only delivered to the organs most affected by their frataxin deficiency?
So these stem cells are basically blood cells, so they are anyways going to circulate throughout the body. But at this point, we see delivery of frataxin in the organs that are affected. We have not analyzed the other tissues to say if some other organ that's not a primary site of target has any other frataxin expression pattern. So we can look into that, but these stem cells are basically blood cells, so it's going to circulate around.
Okay, so I actually have a personal question here in regard to using stem cells with the current climate of what's going on here in America. Are we concerned that we won't be able to continue using them in the future?
So the hematopoietic stem cells that we use here has been in used for other genetic diseases, and it's something that has been widely explored, studied, and its safety concerns addressed over several decades of research. So as of now, the CD34 hematopoietic stem cells are being used for genetic disorders. So it should, I am not sure, but we hope that it's going to help us treat some diseases eventually.
Okay, perfect. Our next question is, how efficient is the CRISPR gene editing step? Are
So,
all cells fully corrected?
Okay, so there are two aspects to that. So we did the CRISPR-Cas9 editing in the patient's hematopoietic stem cells, and we had a ratio of the editing efficiency was somewhere between 40 to 60% of the cells that we tried to edit. And in the mice samples that we work with, we are seeing a reduced efficiency of editing, which could be the innate response of the model itself. So with the mice, we have about 20 to 30% editing, but the what we see at this time is even with the 20 to 30% editing that we see in the mice, we are able to restore the frataxin expression. So we believe that the 40 to 60% that we see on the patients could have some beneficial effects.
Okay, we have another question here in the chat. Can stem cell surgery be used for the loss of nerve function?
So this, I am sorry, I don't have an answer to that, and I can look into it and I'll get back to it.
Okay, and that one is in our chat, not in our Q&A, just so you're aware.
Yes.
The next question is, were you able to detect isoforms of the FXN gene after editing, as there are isoforms starting from intropho1?
So we have not detected the isoforms, but the data that we see here is on the mature frataxin, which is the 14 kilodalton protein. In some of the organs, we were able to detect the full-length frataxin, the intermediate, and the mature frataxin, but that was all we were able to detect. This could be something to do with the antibody that we have been using for the study, and we could try other antibodies to see if we are able to pick up the other isoforms.
Okay, and we do have time for one more question. I know that there is a additional question in the chat, so if you could answer that one as well. Do you remove all of the GAA repeats, or even include more in the in front, or just part of it?
So the GAA repeats that we tried on the patient blood cell line, we were able to remove about 100,000 GAA repeats. And we also had a loss of a little bit of the sequence of the frataxin gene itself. So we did other studies to understand if we lose a small bit of the FXN gene itself, if that is going to affect the way the protein is being produced, or if it's going to affect its function. And with the CRISPR-Cas9 components that we use, we were able to see that it did not significantly affect the way the frataxin is produced. So the maximum editing that we have seen so far is to remove about 1,000 GAA repeats.
Okay, and there are a few more questions in the chat. Liz, I don't know if, because we have more time, you want to go ahead and let her answer them, or are we going to?
Sure, let's do that. We have a few minutes.
Okay, the next question is, most of the behavioral tests showed no significant difference between unedited and edited lines. Is that because of the level of transfer to the in vivo, especially CNS, is not sufficient enough? Or is there a way to enhance efficiency?
So the mice model that we work with right now is the YG8SR mice with the 800 GA repeats in the human transgene. And what we see with the behavioral studies that we have done so far is we do the transplants at 2 months of age and allow the mice 6 months from then. So it's at least 8 months of old by the time we do the behavioral studies. And with these mice model, our behavioral studies has been a little underwhelming compared to the YG8, our original model. So right now we are trying to understand at what point in the age do these mice actually start showing behavioral defects, and at what age does the editing help in restoring these behaviors. So right now with this model, we are still at the stage of identifying when are we seeing the disease starting and when is the editing helping the disease.
Okay, and then our last question is that can the stem cells produce a Trojan FXN variant for delivery to other cells?
That is a very good question and something that we have been exploring. So this is something we worked on in another disease, and it's something that people have been, who use CD34 cells, have been studying for a while on how the protein or DNA or any transfer that happens from the hematopoietic stem cell sourced cells to the organs happen. And there are a couple of mechanisms how they do. So what we understand and what we have shown before is that when these stem cells settle down in the organs of the disease model, they form connecting tubes that deliver the frataxin protein. And that's one way that we have seen so far that's happening. But there are other methods that people have postulated. So at this stage, all we have seen is the tunneling nanotubes that can deliver the protein, but there are multiple other ways because these are cells that are circulating in the blood, and there are other ways of delivering frataxin.
Thank you so much, and thank you to everyone who presented and came. I'm going to go ahead and turn it back over to Liz to close us up for today.
Sure, that was fantastic. What a great start of this series. Everybody did a fantastic job. And so to wrap things, I'd like to thank all the speakers. And we appreciate your hard work and your effort in explaining what you do, your research to our community. Thank you so much, Jessica, for moderating the webinar. Wonderful job. I also want to thank you, Lane Rotten, who worked behind the scenes to organize all the sessions. We are excited that she'll be joining FARA soon, and she really did all the work in putting this together. Just a reminder to keep your browser open to fill out the survey to vote for your preferred presentation. And we will see you again next Thursday, May 12th at noon Eastern time. Thank you, everybody.